DNA is a nucleic acid that contains the genetic instructions used in the development and functioning of all known living organisms and some viruses. The main role of DNA molecules is the long-term storage of information. DNA is often compared to a set of blueprints or a recipe since it contains the instructions needed to construct other components of cells, such as proteins and RNA molecules. The DNA segments that carry this genetic information are called genes, but other DNA sequences have structural purposes or are involved in regulating the use of this genetic information. Chemically, DNA is a long polymer of simple units called nucleotides, with a backbone made of sugars and phosphate groups joined by ester bonds. Attached to each sugar is one of four types of molecules called bases. It is the sequence of these four bases along the backbone that encodes information. This information is read using the genetic code, which specifies the sequence of the amino acids within proteins. The code is read by copying stretches of DNA into the related nucleic acid RNA in a process called transcription. Within the cells, DNA is organized into structures called chromosomes. These chromosomes are duplicated before cells divide in a process called DNA replication. Eukaryotic organisms store their DNA inside the cell nucleus, while in prokaryotes it is found in the cell's cytoplasm. Within the chromosomes, chromatin proteins such as histones, compact, and organized DNA. These compact structures guide the interactions between DNA and other proteins, helping control which parts of the DNA are transcribed. James Watson and Francis Crick have been awarded the Nobel Prize for discovering the shape of DNA, although they didn't give credit to the sole contributor and rightful owner of this prize, Rosalind Franklin. Rosalind risked her life with countless hours exposed to radiation from her X-rays on the shape of DNA, which is known today as Photo 51. This photo clearly shows that DNA turns because of the detail and accuracy of the last few lines. The first step in making of DNA is the uncoiling of the chromosome. First, it uncoils into a small, tightly compacted line and then into a bigger, zigzag shaped one. It then starts to take shape as a round swirl, preparing to be packed with histones and histone H1. The histones are packed with the strand and with the histone H1 molecules, which is only one of the 5 histone molecules. The histones begin to get larger along with histone H1 as they bundle up in pairs of 8. The DNA is finally coiled very, very tightly as it begins to take shape of the well-known double helix. The DNA finally takes shape and becomes the famous double helix strand that we know today.